going to talk about a number of soil-related issues, but also some of the research that we do. In Gail's lab, traditionally, her lab is focused on the ecology, especially soil and plant ecology in grassland systems. And then I came in and said, yeah, but I like row crops and gardens and a number of other things. And it just happens that some of our USDA partners were looking to apply the lessons of the grassland into those systems as well. So I don't know if you can see this because of the contrast in here, but I often, as I teach students about the environment, I like to start off with the idea that humans are having this undeniable impact on the environment, right? And so this is actually a map that NASA put out that shows in the night where all the human settlements are, right? So we can see those light sources there. And the reason why that's so important is that we're really interested in what's happening at that interface of agriculture, ecosystems, and the environment. How are those things interacting back and forth, right? And how can we as humans be better stewards of those natural systems, especially through how we produce food, which takes up about 40% of the land area of our planet is in some way dedicated to food production. And then in an environmental or resource use capacity, we have to talk about things like water, right? Agriculture is the largest consumer of water. And so anything that we can do to help push that system to optimizing those resources, right? And this is one of the big visions or philosophies behind organic production systems, was to get back to this idea of let the solution for the farm come from the farm system instead of pulling resources in from other places all the time. But as Edmund Hillary says, all of these environmental problems are really social problems as well. And so anything that we present about soil health, where the rubber meets the road, is in human decision making, in human priorities, and economic realities, right? About 10 years ago, I decided that I wanted to become a soil guy. And it's been very gratifying that most of the rest of the world decided to care a little more about soils about 10 years ago, right? So, because we recognize that they're the cornerstone of our food production system. So these are just two quotes that I like to mention. You've seen these maybe on Facebook or something like that. Maybe I should share them with you so we can get the word out. But despite all of our accomplishments as a species, we're still pretty dang reliant on soil and rain. We'd be hungry in about 3 months if those things really went away. As Franklin Roosevelt said, the nation that destroys its soils destroys itself. This is why I'm so passionate about this. I think soil health is also national security. It's also global security, right? Hungry people are angry people. We do a lot of discussion about global changes, the future, looking 30 years down the road. Agriculture gets blamed a lot for its part in climate change. Agriculture could actually be part of the solution to climate change. There's about 5 times more carbon in the ground than there is in the atmosphere. So we can start pulling down some of those greenhouse gases and storing them into our soil systems if we'll look at this mixture of methods. So that's why we call the talk, It's Amazing What a Little Dirt Can Do. The unfortunate thing about starting with a soil story is you have to start with what we've done, and that was covered very well this morning. But this map produced by the FAO shows in pink and red, including that large area of the Great Plains in the US, where some of the worst soil erosion and degradation are happening in the world. And what I'd like you to do is, as you look at this map, and I'll flip to a second map here, is compare what those regions are. So look at South Asia, Sub-Saharan Africa, Brazil, Australia, and let's think about those areas that are losing the most soil. Yeah?
Can I ask what the numbers are there? 0 to 100?
So this is a grading system that they came up with, saying how poor. It's like an index. So I couldn't tell you the actual erosion numbers there. Although here's what I like to tell people in a nutshell: every year, if you gather up where all the erosion is happening in one place, it's a land area about half the size of Oklahoma that goes away, right? In little slips and slivers on the edge of ag fields all around the world. But that's untenable to me that we would be losing that much globally. So again, take a look at this map, and I'm going to show you this one, where the dark red represents people who are iron deficient in their diets. Right? There's about 2 billion humans. In some of those same geographic regions where we're losing soil, we're also currently not nourishing the population. And as we project that out, protein, zinc, vitamin A, there's a lot of nutrients that could fit this same question. So connecting soils and human nutrition is a huge part of what we're doing. Here's my last picture to drive that home. These kids are standing, you'll notice, below a line. That line is how tall they should be for their age. If they were the average height of a 9-year-old in the world, they'd be standing at that line. So they are part of this group of 1 in 4 children around the world that are stunted, essentially because they're probably not getting enough protein in their diet, maybe they don't have enough zinc in their diet. Sometimes it's liver damage from things like aflatoxin that cause stunting. But suffice it to say, this connection between the ecology of the soil and the nutrition of humans is really a huge part of what we're into, of what we're interested in. And I put the Sustainable Development Goals of the UN down there at the bottom, because as I look through those, I don't think we can accomplish any of them without a healthy foundation in soil. Even the issues that are related to education or gender equality in the world, those social issues, if people don't have their basic needs met, they often can't make social progress, right? So I think it's very important to mention this. This is the foundation that we're getting at. So we said soil health is hard to define. I mean, there is this definition that's sort of a working definition from NRCS. When I teach my students about soil health, I say, I'm going to give you a really simple definition. Soil health is the opposite of unhealthy soil. Right? That's a horrible definition scientifically. And yet, if you take any physical, chemical, or biological measurement, I can't always tell you what's perfect, but I can sure tell you what's wrong, right? If your pH is 3 in your soil, that's probably not healthy soil. If your physical aggregate stability is low, that's probably not a healthy soil. Can anybody tell me, have we ever found a percentage of OM that's too much? Right? Like, I mean, I guess if we were planting in 100% organic matter, maybe that would be too much. But in general, almost everything falls in these ranges, a high range and a low range, right? And we want to get into that middle and kind of keep the system buffered in that middle range. So as I said, the world is catching on to this. We have Soil Health Day, and the UN declared 2015 the Year of Soils, and a lot of attention is coming around this new groups out there like the Soil Health Institute that are defining and parameterizing these systems. In our lab, we focus on the living part of the soil. So I want you to imagine that you have a paper clip in the palm of your hand. Now turn that to its equal weight in soil in your mind. Just a few little 1 gram of soil. You're holding 100 million bacteria cells when you're doing that with that thought experiment, right? It's amazing how alive, how teeming with life soil really is. And that microbial community is really at the center of so many of those things we would measure and call soil health. It influences organic matter. It influences the way nutrients cycle. That community really drives a lot of the decomposition or structure of the soil. So it's a central concept there. And we like to call these guys our tiny allies. In our lab, we primarily focus on arbuscular mycorrhizal fungi. A picture's worth 1,000 words, and this is some alfalfa that we grew in a greenhouse at OSU. And you see those happy big alfalfa plants in the background, and these sort of sad little plants in the foreground. The only difference there is mycorrhizal fungi. We steamed the soil so it didn't have any life in it. We put back all the bacteria in every pot, but we re-inoculated only those back row plants with mycorrhizal fungi. So we're talking about something that can provide a lot of growth benefit And it really does that. It partners with the vast majority of our crops, of our food, our important food plants, grasses in our and other plants that would appear in grassland systems. And that's because it actually connects to the root of a host plant and helps extend the reach of that root system so that it can forage for more resources in the soil. It goes to a finer scale. It helps it pick up especially phosphorus. At the same time, the growth of that sort of little web through the soil, it stabilizes the soil structure. It helps bind those aggregates together and get more air and water moving through the soil. Caitlin here is holding up the roots of some switchgrass that she grew in our greenhouse. And that's, again, that below ground part of the plant. You can see what absent mycorrhizae looks like, and then when mycorrhizal fungi are present. And again, I like to have people think about if you were holding a coffee cup full of healthy soil, and you took the mycorrhizae out and stretched them end to end, you go across 30 football fields with the length of that, right? That's all jumbled up and connected throughout that soil, that length of hyphae. When I say hyphae, I actually mean this kind of structure here that we can stain and observe on a microscope. So what plants form this relationship? Most of them can't complete their life cycle without this kind of partnership, at least in low nutrient soil. Now, the ones that don't form this relationship with mycorrhizal fungi are your mustards, things like canola, amaranths, so pigweed, right? Brassicas. I mean, I love some cauliflower, but I kind of feel sad when I eat cauliflower sometimes that it never helped nourish a fungi. I mentioned that they extend the reach of that root system, so we have a diagram here that kind of shows how this works. Phosphorus tends to stay put in the soil, wherever it is, whereas nitrogen moves around, right? So that phosphorus, which is further away from the root, it might never get intercepted and drawn into the plant. When you add mycorrhizal fungi, those little microscopic, almost root-like structures stretch out and get the phosphorus, yeah?
Back to the last slide. Green leafy vegetables. Did they all form mycorrhizal?
Some of them do, yeah.
Because otherwise you'd have a heck of a time raising them in aquaponics and the like, correct?
Well, in aquaponic systems, I don't expect mycorrhizal fungi would be very active, right? You're just providing nutrients in an inorganic form. I wanted to mention this about phosphorus. I know organic systems, there are kinds of phosphorus that you can apply, right? Rock phosphate, whatnot. If we look at the world supply of mineable phosphorus, the vast majority of it, and you can't see that probably very well, is Morocco. Like 70% of it, okay? It's not a democracy. What scares me is the Sultan of Morocco won't even let outsiders look at assess how much phosphorus they actually have. So they're like, trust us, we got plenty. And if that supply is disrupted, imagine the input costs, how high they would go for agricultural systems. This is why anything that can help us more efficiently utilize phosphorus is a real important aspect moving forward. Let's talk about commercial fertilizers. Now, I'm talking especially about fertilizer like N fertilizer derived from natural gas, right? The typical Haber process, conventional ag system. The use of those is expected to go up worldwide over the next 30 years to maybe double what we're using today. But we know that about half of what we apply doesn't even get into the plant. It ends up in the ocean, it ends up in the rivers, right? And so we have a scenario where these small-scale farmers, they could be applying, 2 units of nitrogen, and they're actually applying 4, and we're losing that efficiency. This is where we're very interested in can we help plants need less commercial fertilizer or any kind of fertility supplemental supply because of what's going on with soil biology? Can we make more effective partners here? And this is just the term for when you get an aquatic system loaded with all that extra nutrient, and then you get an algal bloom and a die-off of fish. This happens in Oklahoma plenty. that we hear fish kills and such. So garden or food production practices in general that can influence AM fungi, we know that tillage can actually physically destroy it, can chop up that network in the soil. Think about those 30 football fields all rambled around in the soil, and then you run a plow through it, you're cutting it apart. It also changes the composition of the species. Sometimes when we stop tillage, it takes 20 years for those species to really change back to the way they were before. Fertilization, especially over-fertilization, as Gail mentioned, can have a huge influence. It can reduce the abundance, it can change the diversity of what we have going on there with the fungi. How we utilize fallow or crop rotations, this can influence AM fungi. If you grow canola year after year after year, and it's never giving carbon to the fungi, then in that canola field, you're going to have almost no population of these. So if you come back with wheat or sorghum or something that utilizes this partnership, you've just basically eradicated it. And then we're starting to understand more and more research is being done on the secondary effects of herbicides and pesticides and how those might be influencing not just AM fungi, but a whole suite of microbes in the soil. For a long time, we just assumed that if you kill a plant with Roundup, that it doesn't make it into the hyphal system, and there's some research now that says it might be. So we can actually assess soil with a technique where we look at the abundance, not just of AM fungi, but of several of these other microbial groups. So that can give us a snapshot of an idea of our management practices. Are they building the abundance of microbes, or are we losing that? A few years ago, I went to Zambia to visit a friend in the Peace Corps, and I found that every one of the local farmers, as compared to a grassland system right by their farms, their microbial abundance had dropped to about half. These are small-scale farmers. They're not using big equipment. They're not massively fertilizing, but just the process that's going on there, unfortunately, we're losing AM fungi and a lot of our other microbes through the food production systems that we have. So that brings us to this simple question. Could we manage these tiny allies to make a more productive, efficient food system and to rebuild soil health? Can they be managed in these ways? We actually already do this in grasslands. We've been studying AM fungi in grasslands. We know some of the effects of burning or grazing, on these fungi. We know in many cases how to improve their abundance in a system that's been weakened. But to bring that over into agriculture, this is sort of the framework that we've been developing. And this is what makes the research hard. You need to address three things as a system, right? How are you amending your soil? So that whole fertility question. How are you managing your farm? So we've done some research with intercropping versus monocropping, right? That's a farm management decision. Cover cropping is a farm management decision. And then even how are we breeding our crops? Now, I have some good friends that are breeders, so I'm not going to talk really bad about breeders. But sometimes we may have accidentally gone in the wrong direction with the way that we've bred plants and animals. So to have a mycorrhiza smart food system, you would need to bring these three elements together and really understand how they're influencing each other. So tillage is my example. We've known for a long time that tillage can cause a lot of issues with soil biology, right? Introducing all of that oxygen in there, chopping that system up. It can, I don't think I'm preaching to the choir. So, but I see bare soil versus cover crops, as we talked about this morning. Those cover crops are potentially feeding my AM fungi. That bare ground, it's like sitting out in the sun without sunscreen for those poor little guys, right? They're heating up, they're getting nuked essentially. So I'm very positive about reduced tillage and cover cropping as two of the best things for AM fungi. We kind of doubled down on using sorghum as what I would call a model for us. I've become decent at growing sorghum, especially in the greenhouse. And it's really it's an important grain crop, but more importantly, it makes a really good model for some of the genetics that's going on here. So if we can learn some things from the genetics of sorghum, we could potentially move them into breeding for a lot of crops. We could take those lessons and transfer them. And so, again, I don't want to badmouth the breeders. They have delivered disease resistance, drought tolerance, they've improved yield, right? But those are all, for the most part, those are above ground things. They've drastically improved what we can see. But we're asking, what about what we can't see as easily? What about these interactions that are happening right where the roots and the microbes meet? And are we supporting, through breeding, enhancing that partnership, or are we possibly losing that? The way to assess that is to compare a number of different genotypes, everything from those early ancestor sorghums up to those commercial modern sorghums. We grew them in the greenhouse, originally 6 genotypes. I had to put this picture on here because this is a more recent study where I actually got the roots. If anybody's ever worked with sorghum roots, they're very hard to keep intact. So I was quite happy that we actually were able to harvest roots for once. But what we found here, these graphs show root colonization, so we can actually, again, look microscopically and estimate the percentage of the area of the root that's colonized by AM fungi. So when we chart that against protein, phosphorus in the grain, zinc, iron, very similar trends. As you have a plant that's more highly colonized, it tends to produce more grain of higher quality, right? This was what we found, up to an optimum range probably of around 60% colonized. Now my commercial varieties of sorghum were typically colonized more at like 20%. So again, it's the first evidence that maybe we've lost something there in the breeding of these modern varieties. So what I like to tell people is the genotypes that were most responsive to AM fungi, that depended on them the most, in low nutrient soil, they were producing 30% more grain, 30% more protein, zinc, and iron. Some of the very things that we know people are deficient in around the world. The other neat thing that we've learned in the lab, and that's Gail with Nancy Johnson and Yanqi Wu, who does Bermuda grass breeding at OSU, and switchgrass. What we found is that if you make this part of your priority of a breeding program, to breed in lower nutrient conditions to facilitate this relationship through selection, you can end up with offspring that are much more responsive to mycorrhizae than their parents. This is the thing we point to, is we can do it. We can change the way that we breed and bring these little tiny allies into the situation a little better. I got kind of interested in biochar as well and did this one project where we added very small amounts, like 30 grams or something, of biochar per plant and a little bit of worm compost. We mixed them together. What we found here is, comparing it to a routine, what a farmer would fertilize with phosphorus, we saw the root colonization substantially higher when biochar and worm compost were added, and the fertilizer was cut in half. 50% less phosphorus, and yet the yield on the plant was very similar. So we're not sacrificing production to increase the efficiency of the use of that phosphorus. And biochar was one of the reasons. It's basically like putting carbon directly into the soil, right? High quality carbon. And we've got ongoing research. You may recognize these two over here. This is at Lynn's favorite research station, and a variety trial of 15 cowpeas that we did for a few years. And our machine finally works again, so we're going to be processing those samples and figuring out what cowpeas or how cowpeas are interacting with mycorrhizal fungi. So I mentioned the sorghum screening that I did before of 6. Here, as we get to the close of my talk, I just want to mention I'm now doing 50 in the greenhouse. It's kind of insane. Every 2 weeks I have to plant 100 more pots. Every 2 weeks I have to take down 100 more pots. It's quite wild. But that range of diversity, and especially because we'll now be assessing some of the genes that are being expressed in the plants, that's going to facilitate breeders to know what gene might be associated with this partnership, right? What gene or groups of genes? How does the plant change when the mycorrhizae are there, and then we're also growing the plants without? All right, so picture's worth 1,000 words. This was my last takedown, and those are my sorghums in the back that were grown for 2 months in fairly low nutrient soil, right? 10 parts per million phosphorus is not very high. So they grew, right on track. They were about to flower, and then in the front here, you have the same variety of sorghum, the same soil conditions, and no mycorrhizae. Right? So we're gonna get an idea of why is it that these ones, what genetically is changing in these plants as a result of the mycorrhizae being there. Every time I talk about mycorrhizae with the public, I have to point out that what you buy that calls itself mycorrhizae is probably not. We see evidence that they're just putting fish meal or other nutrient sources in there. A lot of times if there is mycorrhizae, it's dead, or it's not a very beneficial species to begin with. We actually did a trial with some undergrads in a botany class. I think we had 6 different commercial inoculum. I wouldn't suggest any of them to anybody. Mycorrhizal fungi are probably there in your soils already. You don't have to add it. It's about supporting it and helping it to flourish in that system over time. Understanding these associations, understanding the implication for breeding in particular, is really important. And it's important not just environmentally, but economically, right? There's the chance here to make more money with fewer inputs. Nitrogen fertilizers represent about half the cost of farming in many operations, for example. So anything we can do to get those resources kind of for free from the microbial world would be great.